Welcome, Senior 5 biology students, to this explainer. Today, we're doing something a little different. We're going to treat the biological cell like a bustling, highly organized Ugandan factory. Why? Because to absolutely crush your UACE exams under the new NCDC curriculum, we need to move way beyond just memorizing textbook facts and step into a true, competency-based understanding of the cell as a dynamic, living machine. Think of it as mapping out the ultimate manufacturing plant, where every single organelle and molecule has a specific, critical job to keep the whole operation running perfectly.
Okay, let's take a quick look at our roadmap. We're moving from the biochemical foundations and enzymes right through microscopy and cell ultrastructure, up to tissues, and finally, exactly how to tackle that UAC assessment.
So, to kick off our micro to macro journey, consider this. How exactly does Lake Victoria regulate Uganda's entire regional climate? It sounds like a massive ecological puzzle, right? But the answer is actually driven entirely by one microscopic molecule. Any guesses?
Yep, it's water. And the secret here lies in water's dipolar nature. See, the oxygen atom pulls electrons much harder than the hydrogen atoms do. So the oxygen end gets a partial negative charge, and the hydrogens are left partially positive. This unequal sharing creates these magnetic-like attractions between neighboring molecules, which we call hydrogen bonds. And because of these strong bonds, water has a ridiculously high specific heat capacity. Just like Lake Victoria absorbs the sun's intense heat slowly during the day to regulate our regional climate, water is doing the exact same thing in our bodies. It's the ultimate temperature regulator, basically acting as the vital coolant for our cellular factory.
All right, let's look at the other raw materials in our factory. Take lipids, for instance, your fats and oils. Think of them as both the long-term energy storage vaults and the concrete walls providing structural integrity for your cellular membranes. They keep the whole factory enclosed. And then you have proteins. Wow, proteins are the versatile structural workers and the heavy-duty transport trucks. From providing structural support, like the collagen in your skin, to acting as defense antibodies fighting off invaders, proteins are literally doing the heavy lifting. So if lipids are the concrete walls, proteins are the specialized doors, the assembly line workers, and the internal transport vehicles, making sure everything gets exactly where it needs to go.
Moving right along to Section 2, enzymes, your biological catalysts. Now, without enzymes, metabolic reactions would happen way too slowly to keep you alive. The really crucial point here is how our understanding of them has evolved. Historically, scientists relied on the older, rigid lock and key concept, where the enzyme's active site is seen as a perfect, unchangeable fit for the substrate. But the modern induced fit model gives us a much more dynamic picture. It shows how the enzyme's active site actually flexes and molds around the substrate when it binds. Think of it like a flexible glove wrapping tightly around a hand. When the enzyme does this, it puts physical stress on the substrate's chemical bonds, which lowers the activation energy needed for the reaction. That is an absolutely foundational concept for your UAC papers.
And you've got to master the specific factors that affect this activity. Temperature, pH, substrate concentration, and inhibitors, both competitive and non-competitive. These all dictate how fast our microscopic machines run. Take temperature, for example. If it gets too hot, the enzyme denatures. It literally physically unfolds, completely destroying the machinery. And why does this matter outside the classroom? Well, understanding metabolic flux has direct, highly profitable applications right here in Uganda. Controlling enzyme activity is the exact science driving local brewing, dairy production, and massive fruit juice processing operations.
Next up, Section 3, Microscopy and Cell Structure. To actually see this factory, we need the right tools.
Okay, I need you to burn this unit conversion into your memory right now. 1 millimeter equals 1,000 micrometers, which equals 1 million nanometers. I literally cannot stress this enough. Calculating linear magnification from a light microscope is a non-negotiable skill for your Senior 5 exams. Seriously, slow down when you're doing this math. If you mess up a single decimal point or forget a conversion, you're throwing away guaranteed marks on your practicals. Memorize this sequence.
When you finally look through that lens, you'll notice a massive structural jump that basically divides all of life. On one hand, you have the simple prokaryotic bacterium. It has no nucleus, it's tiny, just 1 to 5 micrometers, and it uses smaller 7ds ribosomes. Contrast that with the highly developed eukaryotic plant or animal cell. It has a membrane-bound nucleus acting as the head office. It measures anywhere from 10 to 100 micromotors and features larger ADS ribosomes. And a quick expert side note on those ribosomes, that S stands for Svedberg units. It's just a measure of how fast they settle when spun in a centrifuge, giving us a clue about their size and density. The ADS eukaryotic ones are noticeably denser and more complex.
Now pay close attention here, because there's a major curriculum shift under the NCDC guidelines you need to know. What we used to broadly call Kingdom Monera is now officially divided into two distinct domains, eubacteria and archaeobacteria. The catch? This huge division is based on non-structural biochemical differences, like their RNA sequences and specific membrane lipids, not physical traits you can see. Archaeobacteria, for instance, have entirely unique membrane lipids that let them survive in crazy extreme environments, like boiling hot springs or highly acidic lakes. Definitely keep that evolutionary distinction in mind for your structured questions.
Zooming in on the boundary of these cells, we find the fluid mosaic model. Think of this as the highly intelligent security gate of our factory. It describes the plasma membrane as a dynamic phospholipid bilayer with embedded proteins and carbohydrates. Let's break that down. It's fluid because the phospholipid molecules are constantly moving laterally, sliding past each other like a sea of oil. And it's a mosaic because of the diverse, scattered proteins floating within that moving sea. So it's not a rigid solid brick wall. It's a breathable, moving boundary that selectively lets vital raw materials in and waste products out, using passive and active transport.
Moving on to Section 5, Tissues and Organization. Let's zoom out from single cells.
This breakdown perfectly illustrates how individual factory workers team up to form specialized departments. Let's look at animal tissues first. We've got epithelial tissue designed for outer protection, connective tissue for structural support, muscle tissue built to contract for movements, and nervous tissue for rapid electrical signaling. Contrast that with plant tissues. The xylem is like the heavy-duty plumbing system, moving water up from the roots while keeping the plant structurally upright. The phloem, on the other hand, is the distribution network, moving food produced in the leaves everywhere else. And finally, ground tissues like parenchyma, collenchyma, and sclerenchyma handle storage and offer flexible support. It's just a perfect example of cells collaborating to build complex, multicellular life.
All right, on to our final and arguably most crucial section, mastering your UACE assessment. Since the new curriculum is competency-based, you won't just be asked to recite simple definitions anymore. You're going to face scenario-based items exactly like this one. A farmer notices that crops near saline water sources wilt quickly during dry seasons. Explain the biological process responsible. Now, you can't just drop the word osmosis and move on, no way. You have to walk the examiner through it step by step. You need to explain how the highly salty soil creates a hypertonic environment outside the plant roots. Make sure you emphasize the word hypertonic. That means there's a lower concentration of free water molecules outside the roots than inside. This steep gradient physically forces water to move out of the cells and into the soil. As the plant loses water, it loses its turgidity, causing the entire crop to collapse and wilt.
And hey, when you're observing actual specimens through the microscope for your Paper 3 practical, you must strictly follow these rules to secure full marks on your drawings. Number 1, use a sharp pencil. 2, draw large diagrams that actually fill the provided space. 3, and listen to me carefully here, avoid shading entirely. Seriously, no shading. It is the #1 reason students carelessly lose marks. A biological drawing is an outline, not an art project. 4, label with clean, straight lines that don't cross each other. And 5, always include a descriptive title. These rules are absolute, and they will save your grade.
Let's wrap up with this powerful thought. Mastering these concepts develops competencies essential for Uganda's future scientists, doctors, agricultural experts, and researchers. As we finish this explainer, remember that deeply learning these cellular foundations isn't just some hurdle to jump to pass your UNIB exams. It's about building the analytical mindset and practical skills needed to actually solve Uganda's future challenges in health, agriculture, and local industry. You are training to be the factory managers of the biological world. So the question is, are you ready to take those challenges on?